Bismillahirrahmanirrahim. Welcome back, friends. Welcome back to another video lecture by Kami Microbiologist. Today in this video lecture we are going to talk about RNA alternative splicing, or we call that differential splicing, and why we need differential splicing and what's going on inside the differential splicing. And we will talk to the introduction of alternative splicing or differential splicing in a detail. If you guys remember, in the previous video we already talked about the Intron mediated splicing. If you missed this video lecture, it is present into my channel. You can go back and watch it. So let me erase this one portion and let me start the alternative splicing. Okay, so let me write alternative splicing or differential splicing. Okay, now what actually occur in alternative or differential splicing? First of all, let's define it. Alternative or differential splicing is the process in which which mainly occur during gene expression, in which a single gene is give to rise multiple of the protein. So during transcription, when we get the RNA, so this RNA should be spliced. Now the Intron portion are released out, and some of this Intron portion, the Exon portion are also released out, and we get a very short exon connected to each other, which is the mature mRNA. But this single gene give rise multiple of the protein, and this is called as alternative splicing or differential splicing. In this case, different kinds of changes occur inside the messenger RNA. So what kind of changes occur, we'll be talking all in this video lecture. So the first changes in this case, what actually happened in this case, the first alteration is the exon skipping. Exon skipping. So as the name suggests, what actually we have in this case, we have the this exon, as you can see, first exon, and then we have the second exon, and then we have the third exon. Okay, between them we have the Intron portion, as you can see. This blue one represents this blue line represents the Intron portion, and these black rectangle are the exons. Normally what happen, these exon should be removed. Why? Because with the exon portion, all of you better know that exon, in every exon, so let's suppose here we take one of the exon portion. So in every exon we have the 3' hydroxyl group and 5' phosphate group. So what happened, this 3' hydroxyl group have a lone pair which help to attack on the phosphate, alpha phosphate, and this is called nucleophilic attack. Due to this way, the exons are connected with each other. So the same thing will happen in this case. So the Intron are released out. Why? Because in this case, at the mid portion of each Intron, each two Intron have adenine residues or guanine residues, and adenine or guanine residues, they have 2' hydroxyl group. Due to this way, what happened, let's suppose adenine and adenine, so they have also a hydroxyl group which we called it 2' hydroxyl group. So they will attack to this area. So at that specific area we have the phosphate, and similarly the same process will also occur at that specific area. So we will get the larynx in both the cases, and after that the exon will be free, as you can see, and the exons at the end, these are the exons are free and the introns are removed out in the form of larynx, and after some time we will get the structure like that. So this is the normal case. Sometimes what actually happen in alternative splicing, this one exon will be skipped, or this exon will be also removed by this intron as well. So the first intron and the last intron will just connect to each other, and while the mid portion of the intron will be also released out. So this is called as exon skipping. So in this case we have, this is the first exon and the second exon, third exon, which are connected to each other, and then this one exon will give rise protein, different multiple kinds of the protein we will get with this single with this two exon product, not actually two, but with this small genes, with this small region of the genes. This is the first alteration which mainly occur in alternative splicing. The second, we have the mutually exclusive exon. So in this case, mutually exclusive exons will also occur. So what actually happened, let me write: mutually exclusive exon. So now what happened in this case? In this case, we have again the exon. So let me draw here. So we have 4 multiple exon. So let's suppose these are the 4 multiple exons, as you can see, and these black are the intron. Okay. Sometime what actually happen, how the connection will be possible among each of the exon. Sometime what happen, this exon will join with this one exon. Rather it join with this exon will join with the third, and this third will sometime join with the second. Instead they will join with the fourth, and sometime what actually happen, this fourth one will join with the first, and sometime this first will join with the third, except this second. So when this one process will occur, that the exon will connected with each other, but not sequentially. It will just connected with alternatively. So this is called mutually exclusive exon. Okay, so in this case what we have the structure like that. So after the intron are removed out, then we will get the structure like that. So this is the first and with this we have this one. Sometime this, or sometime what actually happened, the second possibility may be like that. So this is the second possibility. So two possibility may occur, and the rest of the exon will be removed out. This is very important. Okay, so in this case the exon will also released out, but sometime this first one exon will react with the second, or this first one exon will react with the fourth one exon, and we will get two possibility like that. And sometime what actually happens, sometime these or sometime this one exon will react with this one portion. Okay, so these are the main possibility that will occur. So this is called as mutually exclusive exon, and it will also occur in alternative splicing technique as well. This is the second changes that may occur in the alternative splicing. Then the third main changes which may also occur, we call that alternative donor site. Okay, what happened in this case? In this case, one of the exon have some specific some specific sites which is acting as a donor, and due to this way it will donate something to the next exon, and due to which the next exon will connect it with each other, and one of the exon will released out. So in this case, again what we have this exon With this exon, we have another exon, as you can see with this red marker. So this is another exon. And after some time, there will be the Intron portion. And after that, we have another exon. Okay. During alternative donor site, what actually happened in this stage? This one exon, as you can see, they will represent something, what we call that hydroxyl group. As you can see, this hydroxyl group will attack with this exon having the 5' phosphate group. So once it attack with the 5' phosphate group, so we will get the structure like that. And remember, this one exon will also be cleaved out. So we will get the structure like that. Let me draw here. This is the exon and this is the Intron. So we will get like that possibility. And this is called as alternative donor site, because in this case this red one represents something to donate, and this is maybe the hydroxyl group. Now the fourth, we have another alteration that may occur in the alternative splicing. We call that alternative acceptor site. So what happened in alternative acceptor site? So it is opposite to this alternative donor site. In this case, what we have one of the exon which is present at the left side according to me, and with this exon then we have a large Intron, as you can see, and after that we have the another exon with this red color, and with this another exon we have another exon with this blue color, as you can see. So what happened in this case? What happened in this case? This one exon will have something to accept. So in this case we have the in this case we have also the hydroxyl group, but in this case we have the phosphate group, as you can see. So it will, so here we have the donor and this is the acceptor. So the donor is this one exon and the acceptor is this one exon. So what happened during this way? It will, during this way, nucleophilic attack will occur. So due to this nucleophilic attack, the next exon will released out. So how this next exon will be released out in both of the cases? In both of the cases, this exon, in this case as well as in this case, it will be released out because between them, a small stretch of sequence is also present between them. As I draw it too much closely, but still between them, a very small stretch of the nucleotide is present. Due to this way, there will be a chances that this one exon will be also cleaved out in both the cases. So what happened? Again, in this case, we get the structure like that. The first exon and the next exon. So we will get the structure like that. And after that, it is ready for the protein synthesis. Another possibility that may also occur in alternative splicing, which is very rare, and we call that Intron. Let me write Intron retention. Now what happened in Intron retention? In this case, we have, it does not occur, okay, but in some cases it may also occur. So we have the exon, as you can see. So 3 exon we have, among them we have the Intron. So during splicing, what happened? The Intron are released out. Okay. So after the Intron, once the Intron will released out, so then the exon will be connected. So let me draw. This is first exon, second exon, and third exon. So how it will be connected? Remember, between them again the intron is present. Although the splicing occur and the intron is removed out, but so not the full intron is released out. Means we can say that 97% intron are released out, but 3% intron is still present among each exon, and this is called as intron retention. So these are the main steps and these are the main stages that will occur in the alternative splicing or differential splicing. Okay, and due to this way we can what we can see in each of the cases, we see that exon should be released out. As you can see, in the first stage the exon will be released out, in the second case the exon will be released out, similarly in this third case we have also the exon is released out. Similarly in this case the exon is released out. As you can see, black and red, but blue is not present. Similarly in this case the intron is present between them, but the exon is also connected. But sometime one of the let's suppose this one, let's suppose intron will be released out, and we have just like that case. So in this case one exon is also released out, but the two exons which are connected to each other, among them a very short portion of intron is also present, so we call it intron retention. So this is all about the alternative or differential splicing. Hopefully you will get idea about that, but if still you have any kind of question, you can write it into a comment. I hope you get idea about that. If you like this, so please make sure to hit the like button and share this video, and definitely subscribe the channel to get more interesting videos like that. Thank you so much for watching.